All right, guys, let's do a quick review of chapter 3. Chapter 3, this was the chapter that was dealing with the tools of microbiology, microbiologists in general. We talked about the 5 I's. The 5 I's were inoculation, incubation, isolation, inspection, identification. So that's the order as well. And you do need to know what the 5 I's are, what the correct words are for the 5 I's, what order you would be doing them in, and be aware of what each of the 5 I's stands for. Inoculation, we are talking about, taking a sample from your patient or whatever it is and introducing it to sterile media in order to hopefully grow it up and analyze it with whatever you're analyzing. The next step is incubation. We want to be sure that we're growing our bacteria at the correct temperature for the correct amount of time. Hey, maybe whatever bacteria you're trying to look at that's causing your patient's, cough or whatever, maybe it takes a whole month to grow and you're just allowing it 2 days and you're not giving it enough time to grow. So that's a possibility. That's something to consider about. Unfortunately, that would be awful to deal with, but it is a possibility. Or like how much is your organism anaerobic, meaning that it can't withstand oxygen, or does it need a certain amount of oxygen or need a certain amount of CO2? Is it a capnophile, meaning that it needs a certain amount of carbon dioxide and that sort of stuff? So that is incubation and how long you're incubating for and the temperature and all of that. Then we have isolation. So we have maybe gone out and swabbed your patient's throat and put it onto sterile media, and we have incubated it. And now we have this whole mess of just a mess growing onto a plate, and you can't get any individual colonies. So what do you do next? Well, one of the things you could do is try to do a streak plate, and that would be diluting out the organisms as best as you could onto a plate, making sure that you are flaming your loop in between and you're not going back and picking up more sample. You're just like, you put it down. No, I know, I learned it this time. I caught it, or I did it. You have your plate, you put maybe you take your actual swab from your sample, or you go into the plate that you tried to culture, but it was too much growing on it. And you take a sample from that. Then you flame. This is a flame. I'm sure you can tell that that's what that is. You're gonna flame your loop, and then you're going to go in and squiggle, and then you're going to do it again, and you go in and squiggle, and you do it one more time, and you go in and squiggle, squiggle, squiggle all the way out. And eventually, because we didn't go back and get more sample or anything at this point, you should have single colonies. Remember, a single colony arises from one cell that has grown up. So then you could potentially take that one colony and put it into sterile media. And if you're like, hey, I'm not really sure, there's a couple things growing here, which one do I want to look at for my testing? Then take both of them or through all of them, whatever you think you need to do. That would be the next step. So right. So let's say you get your individual colony and you go and you put it in a new media and it's going to grow and that's fine. That would be called a pure culture. So that's a term to be aware of. And anexic is an even more specific term about it, where we're saying it is free of growth except for the target, whatever you're trying to grow, right? So those are pure culture. There's different kinds of media that you can grow your sample on. Selective media, that's going to prohibit, it's going to have something in it that's going to keep other things from growing. So only certain kinds of bacteria can grow, like gut bacteria, when we add in bile salts to McConkey agar, for example, or whenever we have salt that we add in to mannitol salt agar, in order for us to grow, skin-based bacteria like Staphylococcus organisms. Then we have differential media. This is media that will allow for like color changes or something based on whether that organism can metabolize something in the media. Like with mannitol salt agar, if it turns yellow, Then the original color was a pinkish red. So if it turns yellow, that means that the organism can metabolize the sugar and make an acid byproduct, and it changes color based on the pH change. So that's an example of the differential aspect of that media. And we already talked about that. That one is also selective. So it can be more than one. Then fastidious media or fastidious organisms. Fastidious organisms grow on enriched media where we have added a growth factor. What all that means is fastidious organisms need a growth factor. A growth factor is something that the organism cannot make on its own, and it has to be supplied. Like we need vitamin C or a lot of our vitamins, like B12 and stuff. We need that in our diet. Okay, so that was talking about isolation. Now when we go inspection, one of the ways that we inspect our microorganisms is using microscopy. I want you guys to know the different parts of the microscope. I'm not going to go over them here. This is something that you guys should be able to do on your own time. The different kinds of microscopes. We talked about light microscopes. We've talked about, we didn't get too much into talking about the electron microscopes, but electron microscopes are a thing as well. They give us a lot more resolution than everything else. They are actually going to give us the most detail of all of them. So that's what electron microscopes are for. Microscopes, right. So they're going to magnify. You're going to get your real image in your objective lens, and then you're going to get your virtual image in your ocular lens. So that's how, based on how the light is traveling out and when magnification is happening of your actual specimen image. Oil. We add oil at our 100x on our own microscopes and our light microscopes. The oil is there to prevent light scatter and improve the image. So we can see things a little bit more clearly because the oil and the glass of the slide, as well as the lens itself, those have similar properties. Whereas with the air is considerably different from the glass. So it leads to light scatter. So the oil will reduce that. Then we have introduction of different stains. And we talk about specimen preparation. You want to decide on your specimen preparation based on whatever you're wanting to look at about your organism. If you're wanting to look at like motility, for example, you might want to do a wet mount or a hanging drop mount. If you're doing a stain, then you might want, for most stains, to do a heat fixed smear. For our lab, we do not heat fix our capsule because the heat fixing can actually cause the cells to shrink and make it look like you have a capsule when there really isn't one. So that's why we do that. Yeah. Yes. So gram stains that are allowing us to determine the differences between the cell envelopes. The gram positives are purple and gram negatives are red in that stain. Acid fast stain, looking for presence of mycolic acid in the cell envelope. That's going to be the case with Mycobacterium species. So positive for that one is going to be pink fuchsia. The stain we use for that is carbofuscin. And then we have negative cells will look blue in that because we will counter stain with methylene blue. Endospore stains will allow us to see the difference between vegetative and endospore cells. Vegetative cells and the endospores, which are these protective structures that cells make to protect their genetic material in harsh conditions. That will include genera Bacillus and Clostridium. And then we have capsule stains that will identify bacteria that produce capsules, which are going to be for evading phagocytosis, which is very important in pathogenic organisms. And then we have flagellar stains. So it'll have stains that actually dyes will deposit onto the flagella themselves to make them visible. So that can be another defining characteristic about some microorganisms is how many and where their flagella are located. So that is it for chapter 3 and the rundown for that. And it's been fun, but I'm going to go ahead and cut you off here and I'll see you guys for the next one.